Good evening, everyone. I am Tanisha Biswas, postgraduate student of Department of Physiology, Srirampur College, affiliated to the University of Calcutta, West Bengal, India. Welcome to all in today's webinar on research methodology organized by South Asian Center of Excellence in Physiology, Education and Research. India is organizing the webinar this time. Without any further delay, I would like to introduce Dr. Kaushik Bharati. It's my privilege to introduce you, sir. Dr. Kaushik Bharati is chairman of the SAAP Research and Publication Subcommittee, editor-in-chief of the SAAP Bulletin, and is also involved in developing the SAAP Journal. He is currently a health policy consultant at UNESCO and a former consultant at WHO. He did his PhD in physiology from the Calcutta School of Tropical Medicine and postdoctoral fellowship from the Liverpool School of Tropical Medicine, UK. He is the Vice President of the Physiological Society of India, elected member of the Infectious Diseases Society of America, Arlington, Virginia, and American Society of Clinical Oncology, Alexandria, Virginia, and a Fellow of the Royal Society for Public Health, London. We are honored to have Dr. Kaushik Bharati with us. I now request Sir to deliver the opening remarks. The platform is all yours.
Thank you, Pranisha, for the nice introduction. Uh, Unfortunately, our president, Professor Samila Malik, will take some more time to join. So I welcome Professor Naima Sultana, our Secretary General. A very warm welcome to you, and the speakers, of which I can see Dr. Aniruddha Roy. Welcome, Aniruddha. And Arnab Banerjee, he is also a speaker. And this is being organized by the South Asian Center for Excellence in Physiology and Education and Research, SACE-ER. And this is, we have two arms of the SAP. That is, one is the SAP Education Subcommittee, and the other one is SAP Research and Publication Subcommittee. And we will be organizing these series of webinars this year and the following year till December. And it will alternate between the Education Committee and the Research and Publication Subcommittee. The last in June 17th, the first webinar was conducted by the Education Subcommittee on the topic innovative techniques in teaching and learning. So that was a very good successful webinar actually. And today we are organizing and focusing on the topic of research methodology. And we also plan to take this forward and as I mentioned, till next year. So I hope you will be able to support us and by joining and giving us support in this regard. And I'm very glad all of you could be able to join. It is especially heartening to know the students are joining. They are the backbone actually of our community, actually student community. And so without further ado, I'll give the floor to Pranisha to conduct the rest of the webinar. Thank you very much.
Thank you, sir, for your inoculant speech. Now I would like to introduce Professor Naima Sultana. Professor Naima Sultana is Secretary General of the South Asian Association of SAAP, Physiologists. She is a highly regarded physiologist with over 31 years of experience in medical education and government medical colleges, both in Bangladesh and abroad. She received her basic medical science degree in physiology and her MSc in physiology from the Institute of Postgraduate Medicine and Research, affiliated with Dhaka University. Subsequently, she attained her MPhil in physiology from Bangabandhu Sheikh Mujib Medical University. Throughout her career, Professor Sultana has held positions at several esteemed institutions, including Sir Salimullah Medical College and Manikganj Medical College in Bangladesh. From 2017 to 2022, she taught human physiology at the University of Papua New Guinea. She has actively contributed to undergraduate and postgraduate teaching, curriculum development, and research supervision. Her research interests encompass micronutrients, enzymology, nutrition, hematology, and cardiorespiratory physiology. Professor Sultana has published extensively in both nutritional, national, and international journals and presented her findings at global conferences. As a devoted educator, she is committed to promoting excellence in medical education and research. Currently, she also teaches online classes at the School of Medicine and Health Sciences, University of Papua New Guinea. We are honored to have Professor Naima Sultana with us. It is my earnest request to ma'am to say something on today's webinar.
Thank you, Dr. Pranisha, for your kind introduction, and thank you, Dr. Kaushik, for arranging such a wonderful session. Good evening, everyone, and all the respected speakers, distinguished participants joining today's session. It's my great pleasure to address you at this important webinar on research methodology organized under the auspices of South Asian Center of Excellence in Physiology, Education and Research, that is SACE-ER, and led by the Research and Publication Subcommittee of SAP. I would like to extend a special note of appreciation to our speakers for graciously accepting the responsibility of delivering this insightful session. At SAP, we firmly believe that quality research is the foundation of scientific progress. A sound understanding of methodology is essential, whether we are designing experiments, collecting data, or interpreting results. This webinar is not merely a discussion, it is a valuable capacity building opportunity for young researchers, early career academicians, and all those committed to advancing physiological sciences. I take this moment to sincerely acknowledge the dedication of our Research and Publication Subcommittee led by Dr. Kaushik. Your enthusiasm and tireless effort in conceptualizing and organizing this program are truly commendable. We are privileged to have three distinguished speakers with us today, each bringing a wealth of knowledge and expertise. And I encourage all participants to engage actively and make the most of this session. In conclusion, I extend my heartfelt thanks to all our speakers and participants. Let this be the beginning of many more collaborative efforts to elevate the standard of physiological research in our region. Thank you all.
Thanks a lot, ma'am, for your nice talk. Now it's the time to introduce with our eminent speakers. Our honorable first speaker is Dr. Arnob Banerjee. Dr. Arnob Banerjee is resource person at the Postgraduate Department of Physiology, Srirampur College, affiliated to the University of Calcutta, West Bengal, India. He is also associated with the Department of Food and Nutrition at the University of Kalyani, India, as a guest faculty. He is an accomplished physiologist with over 13 years of experience in research and teaching background in biological sciences. Dr. Banerjee completed his PhD from the University of Calcutta, then moved to the US for his postdoctoral research at the College of Medicine, Department of Pharmacology and Nutritional Sciences, University of Kentucky. He is an expert in the field of cellular biochemistry and molecular biology. His research is mainly focused on metabolic disorders and reproductive biochemistry. With an age index of 13, Dr. Banerjee published his work in different international peer-reviewed journals with high impact factor, including publishers like Nature, Springer, Elsevier, Wiley, Taylor and Francis, De Gruyter, and Sage, among others. He serves as an editor and reviewer of different international peer-reviewed journals. He is also a member of national and international societies, including the Indian Science Congress Association, the Physiological Society of India, Society of Biological Chemists India, International Association of Neurosciences, International Association of Oncology, Association of Pharmaceutical Research, among others. Dr. Banerjee received numerous awards from different national and international societies, journals, and web of science. Dr. Banerjee will speak on the topic cell culture in biomedical research. The platform is all yours, sir.
Thank you, Pranisha, for your kind introduction, and thank you to all of you, our respected organizing committee member, for inviting me to share my thoughts on the cell culture. So, my slide is visible.
Yes.
Okay, thank you. So, before going to details in the cell culture, I would like to share that the cell culture is a cornerstone of the biomedical research that enable the disease modeling and drug testing and breakthrough in personalized medicine, that making it essential for the robust research methodology. So we are all ultimately running toward the cell. It can be either from the genetics or molecular biology, or in the case of the immunology or microbiology, our main goal is the cell, and that converges ultimately at the cell or cellular level. That is particularly the fundamental unit of the life, and the research mainly focus, and we are all running towards the cell. So first we have to know that what is a cell. So we already know that the cell is a fluid, and particularly a fluid filled and nutrient rich material, just like a balloon, and that is the building block of our body. So a cell can be compared with a balloon that is filled with a nutrient rich molecule or nutrient rich solution in that case. So if you look at the cell is building block of the living organism, and cell, we know that the cell is the basic unit of our life. So in that case, it seems estimated that the human body contains nearly about 37 to 38 trillions of cell approximately. And here this photograph shows that the specific type of tissue and one more tissue work together to form an organ in that case. So before diving into the research, we must know about the difference between search and research. So in that case, which is already published or from the existing knowledge of literature, and we should identify the gap before doing the research. So first is the research, that is the explores the existing knowledge and identify the gap and analyzing the past result, and only then can true research begin bridging those gaps with the innovation purpose. So before going to start in detail in the cell culture, you have to know that what is the history, that is the search. So in 1951, Henrietta Lacks, a black tobacco farmer, unknowingly donated her cancerous cell, that is HeLa, to the science before dying at 31 age. So unlike other cells, HeLa thrived indefinitely, becoming the first immortal human cell line, and without her family consent, this cell revolutionized in particularly the medicine and enabling the polio vaccine, cancer research, IVF, even in the space physiology also. And though her story remained hidden for the decades, the Henrietta's cell fueled a multi-billion-dollar industry while her family struggled in the poverty. So today, HeLa symbolize both the scientific progress and the ethical injustice in sparking the global debates on the consent on, and also in the case of the equity in research. So her legacy lives on in the labs, laws, and also the 10 billion HeLa cells still dividing the world. So in that case, we particularly focused on the HeLa cell, and we have to, I think we have to know regarding who is HeLa and who is particularly the HeLa term should be coming from the Henrietta Lacks. And this is the story before doing the research. So in that case, if you wish to know more about the Henrietta Lacks and more about the cell, the basic search for the cell, then you have to know this lady. So this is the Henrietta Lacks, and where ultimately this operation occurs in the John Hopkins laboratory. And this is the short video of the HeLa cell line, which is divided in particularly the 72 hours or 46 hours, in particular this time span. And if you wish to study more about the HeLa cell or Henrietta Lacks lifestyle, I just share my some books, particularly that is the Immortal Life of Henrietta Lacks. And this is another book you can also read to know about more about the history of the HeLa cell line and other cell line also. The main base on the cell culture, that is the conspiracy of the cell. So the story of every single cell, we should focus on that purpose today. So from the lab and how this kind of cell culture and which is and particularly involved in the biomedical research, we need to know that. So the cell culture brings the lab discoveries of the real world that cures the enabling the vaccine, cancer therapies, and regenerative medicine through particularly to scale up and ethical research also in that case. So in exploring the cell culture method for scientific significance, the cells grown in artificially in the nutrient-rich media and under the controlled temperature, humidity, and also in presence of the gaseous substance, either it can be CO2 or other gas that is specific for the cell line. And this primary cell culture or the cultured cell extracted from the tissue that can be either extracted via the enzymatic degradation or mechanical, but mechanical sometimes mechanical is stressful. So in specific cell line, we generally use the enzymatic degradation for isolation the primary cell and grown until they cover the substrate particularly. So the primary cell culture limits a finite lifespan due to the nutrient depletion. And also in that case, if you look at the secondary cell culture, that repeated subculturing and creates the stable cell lines for long-term research or for the industrial purpose. So secondary cell culture is particularly the large volume expand. We can use bioreactor in that case also for large culture for particularly the vaccine production and antibody production also, and particularly the few experiment. Okay. So in that next is what is the application? The basic application of that cell culture studies, that the growth behavior control setting, and all this ultimately reached in the nutrient media. So we should focus on the behavior of the cell, cellular growth, and properties that control the condition. So the significance in that case, in the case of the cell culture or growth behavior under the control setting, advances the molecular biology, genetics, and also in the case of the biochemistry research. So scientists particularly nurture the cell just like a pet. Okay. So they particularly tweak the nutrients for the growth and test the toxin for the safety and edit the gene that particularly crack the disease mystery. So we should focus on the how this cell is growing into the culture media and all these factors, that means the temperature, the humidity, and all this, we should focus on these factors also. So why this cell culture is essential in that case? So if you look at the cell culture, why it is essential and what is the rationality of cell culture in that case, so that particularly enables the drug testing, or it can ultimately focus on how we can focus on the research before doing any in vivo experiment. So this is the first trial. In that case, then we can go with the in vivo experiment in the case of the rat or mice, and then we can transfer or then we can do the clinical model, that is the human model. Okay, so this is the step, or this is just like a staircase of the research in that case. So in that case, we can divide the cell culture into two ways. Okay, So how this two-way, the cell culture media is very much necessary for this kind of purpose, and we should focus what are the cell culture media and how this cell culture media is particularly used in the different cell culture model, and the cell is healthy or not. Okay, so before doing any cell culture, we should focus on few tidbits. So the first is the aseptic environment. Second is the preparation of the growth media. That means the cell food, particularly for the layman. And I will definitely discuss in scientific language, particularly for the researcher. So this is just a food for the cellular growth and to maintain the cell in a culture media outside your body. So creating appropriate culturing environment, that is either it can be 37 degrees centigrade or 34 degrees centigrade, that is particularly depending on specific cell line. Okay, and taking the cell regularly. Next, subculturing the cell for different purpose. You can store it, you can run experiment or splitting it for your experimental purpose. And if you wish to detach the cell, because there are two kinds of cell in the case of the cell culture. First is the adherent cell that is totally attached with the plate or the petri plate or the culture dishes, and another is the suspension cell. So in that case, the subculture attached cell line requires the trypsinization, that is the enzymatic treatment for treating out the cell, and subculturing the suspension cell line, and then changing the media also, because the cell itself release some excretory material, some metabolites that can also change the environment, that means the pH of the culture media. So you should change the media in that case, and you should mark the passage number, how many times you passes that cell. So first is before going to the cell culture room, you first turn on the UV light of that particular room for 20 minutes, then entering the room and then turn on the laminar flow, the UV light for 20 minutes, then turn on the blower that is particularly protected with the HEPA filter for any kind of contamination, and then the gloves, and then wash your hand with 70% alcohol, particularly for the sterilization purpose, and you should be careful about the contamination. So that's why this precaution you should take care before doing any cell culture, and all this cell culture media should be sterile. And after doing it, all this kind of matters, then you can start the cell culture, or you can enter into the cell culture room. So what is the media? So in that case, if you look at we can divide this kind of the cell culture media into two parts: the natural and the artificial. So in common folk, particularly for the common folk, the cell food with nutrients and vitamins and minerals to help the cells grow and stay healthy in the lab, that is the cell culture media. But for us, that is the budding scientist or the researcher, this is the precise nutrient formulation, that is the basal media with the supplement like FDS or other growth factors, that maintaining particularly the pH and osmolarity and sterility, particularly the optimal cell viability and the function. That is the cell culture media in that case. So the purpose in that case is particularly to maintain the pH and the osmotic pressure, to maintain the optimum temperature in that case. And the criteria for selecting the cell culture media, that should be the immediate survival and prolonged survival also for long-term research or from step-by-step research, and indefinite growth, and also specialized function, because every media has some specialized nutrient, specialized composition. So we can divide this cell culture media into two groups. First is the natural, another is the artificial. So in that case, the natural media further divided into three groups. First, biological fluid, tissue extract, and the clots. So the tissue extract like liver, spleen, and leukocyte, and all these are particularly grouped into the tissue extract natural media. Biological fluid you already know, that is the plasma, serum, or lymph, or human placental cord like that, and clots like coagulant or plasma. But what is the artificial part? Artificial is like the balanced salt solution and basal media and the complex media. So if you focus on the balanced media, that is the phosphate buffer saline or the DPBS or HBSS like that, and basal media like modified Eagles media or Dalbeck modified Eagles media, and the complex media like RPMI 1640 and IMD. And there is another part of the artificial media, that is the serum containing media. Serum media offers the growth factor, and serum free reduces the variability. So chemically defined ensure the consistency, and protein free minimizes the contamination risk. So these are the advantage of the serum media and serum free media like that. So this basal media is the another part that contain the keto acid for the energy like that, and some other carbs like the glucose and galactose, and vitamins like B complex and other vitamins you can supplement it, and also some stress minerals in that case, and for the metabolism and growth. And also phenol red is present in the cell culture media to indicate the particular pH change. So if it drops or if it's alkaline, in that case for the cellular, from particularly the cell metabolites or like that, they can ultimately change its color. So when it ultimately changes its color, then you can identify, or you ultimately get alarmed that your cell culture media is changing the color. So that means something is happening. It can be either contamination, it can be either due to any kind of overpopulation of the cell, that is the confluency, or the cellular debris. Okay. So these are the component I just told you about the complement, and then another is the supplement. What are the supplement? The supplement is basically very much required for this purpose, like the L-glutamate, non-essential amino acid, growth factors like hormones and insulin, some kind of hepatocyte. If I particularly do research on hepatocyte, primary hepatocyte, and cardiomyocyte also, so they particularly need the insulin for the growth factor and for the addition purpose in that case. So the another part is particularly the growth factor. What is this growth factor? The growth factor is important in case of the cell culture, because if you wish to culture a cell, like the growth provide, it particularly provide the growth of that cell. So the proteins for the cell that attach particularly with the cell, and we need to heat inactivate it. So how? The growth factors or hormone that particularly aids the attachment of the cell, and that binds to neutralize toxins or neutralizes the toxin, and they are particularly used as some kind of infectious agent. But before adding any fetal bovine serum or fetal calf serum into your cell culture media, you need to heat inactivate it because of the destruction of the complement and the immunoglobulins. But overheating can ultimately damage your media or the serum culture. So the preventing the contamination, you need the antibiotic in that case. Sorry. And in that case of the antibiotic treatment, it can either you can inhibit the cell wall synthesis via the inhibition of the DNA synthesis, or it can inhibit the protein synthesis also in case of the mammalian cell culture. And in case of the bacterial cell culture, which I discuss later, you can use some ampicillin or penicillin or kanamycin, the specific antibiotic for the cell. So in that case, this is called a zone of inhibition. That is, the growth is in the outside, and when the antibiotics, is particular antibiotic plate is particular disc is particularly placed on the culture media, the growth is inhibited in that case. In the case of the bacteria, but in case of the cell mammalian cell culture, you can use the penicillin, streptomycin, that also inhibit this kind of contamination, like bacterial contamination or like that. So the media which mimic the human body fluid, that is natural behavior in that case. So before culturing, you should know about the cell and the specificity of the cell line with their medium. So this is the table which ultimately depicted that the HeLa cell is the epithelial and species definitely from the human, and you need the modified Eagles media with the supplementation of the glutamine and 10% FBS. So this is the growth factor in that case. And other cell line also available in the table, and that particularly applicable in the tumorigenicity or virus study or other kind of cellular molecular biology related study. And these are the common media and their application. In that case, you can use the modified Eagles media or Dalbeco modified Eagles media. Particularly, this media is very much essential for specific purpose. So if you choose that the DMEM, that is the high glucose DMEM, this is for the specific for the cancer cell. If you choose the RPMI 1640, you can use immune cell like splenocyte, thymocyte, or like that. If you choose the MEM or modified Eagles media, this is the basic growth in that case. So everyone has some specificity. So the science of pH stability in the cell culture media, you need to know about the basic process of the mechanism by which the CO2 bicarbonate buffer system is regulating the basic media purpose. So the DMEM and all the other media is particularly in the cell culture. The pH relies on a CO2 bicarbonate balance. The 5% CO2 maintained at 7.4 pH by forming carbonic acid. So in that case, the preventing the alkaline drift and the atmospheric gas exchange, but over ventilation, it can rises the pH. And if the rises of the pH occurs, then the cell gets stressed. So in the next case, what is a contamination in the cell culture? So if you're doing research in the cell culture lab, the contamination or the term conta is very much applicable for you who are particularly in the research field. So what is this? The cell culture contamination are of particularly two types that include the bacteria or fungi or kind of virus, and another part is the chemical. So these two types is first is the biological and another is the chemical. So if you look at this cell, this is the bacterial contamination and these are the main cell line or cell, and this is called the mycoplasma colony. And this is the debris, that is the degraded material in that case. So if you look at this cell culture, this is just 1 to 2% contaminated culture. If you look at this is the bacterial contamination occurs and some kind of mycoplasma also available in that case, and this is fully contaminated culture. So if one bacteria or like one cell is contaminated, then the whole cell you need to just discard this cell. Okay, so the detection is very important in that case, the detection of the contaminants. So general sign of contamination, already, as I told you, the turbid culture. Why this turbidity occurs? Because the changing of the pH. So abnormal pH, you cannot measure the pH by placing electrodes or placing a pH paper or like that. You just check the color of the media. Okay. So poor cell attachment also cause some kind of contamination. And if you look at the visible effect of like the yeast and bacteria, fungi, I already shown you that some kind of visible effect also occurs in the case of the bacteria or any kind of fungi. That also changes the pH. So what is the method? The method is either you can stain the cell or the particular culture with the DNA staining, or you can perform the PCR for the mycoplasma RNA identification, or there is some mycoplasma detection kit also available in the market. You can also use this for detection of the contamination. Okay, so the best method to eliminate the contamination, you just discard the old cell line or the old cell culture, just discard it into waste pot. Okay, and also before doing that, you also regularly check the cell every day, and whenever you get more cell or confluent cell but which are pure without any contamination, you store it in a minus 80 followed by minus 130 and also in the liquid nitrogen. But what are the plate where they are ultimately cultured? These are the cell culture containers, that is the disc. These are called the T25, 75 and 175 flask. These are the 6-well plate and Petri plate, and this is the spinner flask where we can culture the cell, and this is the bioreactor. We can culture large amount of or bulk amount of cell culture in that case. So these are just general purpose of the cell culture containers in that case. So cell culture incubator or the grown flasks or dishes is the one part, and regularly medium change and waste removal, maintaining the cellular health and sterile technique of the hoods and HEPA filters, and all this you need to know about the laminar flow and the cell culture incubator in that case. So this is the CO2 incubator in that case. You need say 37-degree temperature and 5% CO2 supply for culturing many kind of cell, but not in all cases. Because if you culture the Leydig cell, then you need 34 degrees centigrade with the 5% CO2. But in other cases, mostly all cases, you need 5% CO2 with 37-degree temperature incubator. Okay, so that incubate, particularly the incubation is occurs under controlled temperature with controlled atmosphere. Okay, so this is the aseptic condition. What is this aseptic condition? This is the laminar flow where you can ultimately culture the cell. Okay, so this is the overview of the laminar flow. This is the schematic diagram. This is the place of the HEPA filter, which ultimately filtered all the contaminated air, and this is the pre-filters also in there, and this is the workplace where you can ultimately. This is the workplace. This is that. Okay, so this workplace is particularly for the cell culture media, but mind it, the bacterial cell culture and mammalian cell culture, you cannot use the same laminar flow, same culture hood or same culture room for both. Okay, otherwise the contamination occurs. There are separate room for bacterial culture, for plant culture, for insect culture, and for the mammalian cell culture in that case also. So after doing every sterile process like the UV turn on, laminar flow blower turn on, after turn off the UV, you should turn on the blower, and then you sit on a tool with your all accessories, and this is the all accessories for the cell culture media, pipette and culture media are all like that, and then you can perform your cell culture experiment. Okay. So the housekeeping in the cell culture is very much important. And if you look at the aseptic technique, the sterile workplace, that is the cell culture hood, you should clean all these things because otherwise you can contaminate your cell culture. And the next day you cannot perform any experiment. Okay. So cell culture hood and air filtration, that is the HEPA filter, the efficacy is 99.9% particularly, and this particular filter size is 0.3 micron for the particle. And cell culture laboratory safety, this is very much important before doing the cell culture, because we are particularly discuss about the basics of the cell culture. So this is the some kind of rule of thumbs like that. And typical cell culture room prevent the contamination and protect the researcher by containing some kind of biohazard and ensuring the sterile condition for the cell and safety for the personnel and the environment also. So in the CDC that defines particularly the biosafety levels, that is the BSL1, BSL1 and BSL4 for the lab based on the agent of risk that governing particularly the contaminants, and also the personal protective equipment procedure to protect the workers and the environment from the pathogen. So if you look at the BSL1, that is low risk or non-pathogenic condition. If you look at the BSL2, that is moderate risk and human derived cell also. In the case of the BSL3 is also next moderate as compared to the BSL2. But the BSL4, particularly the deadly pathogen like Ebola or like that. So these are the stages of the biosafety as per the CDC. Okay. So in that case, now you can perform the cell culture. Now you can go or you can enter into the cell culture experiment. So it can be either from the animal or it can be from the plant. You just collect the tissue, okay? And then you can get some kind of single cell like either enzymatic degradation or mechanical degradation. Then the secondary culture occurs. And before in between the primary and secondary culture, this is called the subculture. And then after the secondary culture, you can generate the cell line. So this is also a subculture, secondary and cell line, in between the secondary and cell line. and you can store the cell line in minus 80 and then minus 130 and followed by the liquid nitrogen. Otherwise, if you place directly the cell line onto the minus 130, then the cell gets stressed. Okay, so step by step. And the cell line, you can continuously process the cell, you can store the cell, you can run different experiment. And the other thing is the primary, secondary, and the continuous culture. So what is the primary cells in that case? So primary cells taken directly from the tissues closely mimic in the in vivo conditions for the accurate research but have limited division. Okay. So in that case, in the case of the culture, restrict the long-term use also in that case. So cell lines particularly are lab cultured immortal cells from the cancer or transformation that divide indefinitely, and unlike the normal sensing cells that enable the long-term research and industrial application also. So if you look at the immortal cell line and transferred cell line, you can get that this is just a cartoonistic view or the animated view of the HeLa cell, how the HeLa cell is isolated without her concern, and the division rate is high in that case, and this is still immortal. And that's why the HeLa cell is known as the immortal cell all over the world in that case. Okay. So the pluripotent stem cell like cell line particularly, so that divides indefinitely but naturally, not via any kind of transformation purpose. So likely the primary cell, they retain normal traits and can differentiate into different kind of cell type. So if you look at the stem cell, what they are doing, they can ultimately create muscle cell, that nerve cell, blood cell, and other cells also. So pluripotent stem cells are the type of stem cell that can differentiate into any cell of your body. So this is the types of pluripotent stem cell. This is the embryonic stem cell, and this is the induced pluripotent stem cell. So there are particularly two main types of pluripotent stem cell. So the embryonic stem cell, which derived from the inner cell mass, the blastocyst, during the early embryonic development, and induced pluripotent stem cell, which are adult cells, particularly have been genetically reprogrammed to behave like a pluripotent stem cell. So this is just a normal video of the normal cell line versus the cancer cell line. So this is the normal growth rate of the normal cell that is not cancerous. And if you just jump to the cancer cell, in case of the breast cancer, so you can look at the difference, right? So this is rapidly. So they are particularly rapid. The growth is rapid in that case. Okay. So this is called uncontrolled cell division, and the cell growth rate is high in that case. So the primary cell culture, if you look at the cell are isolated from the young animal tissue or fluid or the mesenchymal or any kind of mechanical stimuli like blender or chopping out the cell, or the enzymatic degradation or like trypsin and collagenase also in that case, that is the dissociation of the cell from the matrix. And DNase also prevent the clumping, and selected cell are then cultured in a specialized media, that is they are particularly specific for specific media like DMEM or RPMI 1640 like that. So the primary, this is the overview of the schematic representation of the process of primary tumor cell line establishment. So in that case, the primary neoplastic mass, if you look at the neoplastic mass, this is solid tumor, is dissociated either by the enzymatic or chemical or mechanical degradation or dissociation method. So the tumor cell enriched from the single cell suspension either can be divided into 2D or 3D or explained precision cut like the, and also sandwich or cancer stem cell isolation methods. So these cells are cultured in the medium by adding sustainable tissue-specific supplement in that case. So then you can culture, you can ultimately establish the cell line. This is not a single day research. You can get with the trial and error method, and then you can establish the cell line that is pure. That means without any contamination. So in the primary cell culture, I just showed you that these are from our lab, uh, that is from Department of Physiology, Dr. Bipin Kumar Majhi's lab. This is the hepatocyte isolated from the young adult rat. This is the cardiomyocyte and the splenocyte, and this is the embryonic stem cell recently isolated and cultured, and these are the established cell lines. So if you look at the secondary cell culture, or secondary cell culture, how you can distinguish between the primary and secondary. So you first know about how this cell culture ultimately occurs in the different from the normal cell. So the normal cell line, that is in that case the rat cardiomyocyte, if you look at this is the rat cardiomyocyte that can spontaneously immortalize, and others artificial immortalize like SV40T antigen or derived tumor, that is the HeLa cell line, and hybridoma, that is specialized in research application in that case. So the secondary cell culture, this is the flow of the secondary cell culture. You can particularly get the cell tissue culture from the primary cell culture, and then you can establish the cell line. You can isolate the single cell or desuspend the cell for the clonal selection and immortalize cell for any kind of transformation, or you can store or preserve this cell in a cryovial and successive subculture for any kind of experiments or like that. So this is the cell morphology vary depending on the cell type. So what are these? This is particularly the fibroblastic, epithelial, endothelial, and neuronal cell. These are the main morphological overview of the different kinds of cell. So if you look at the adherent and non-adherent cell, this adherent and non-adherent cell means that is the adherent cell that attach to with the cell surface. That is like fibroblast or HeLa and requires proteolytic enzyme trypsin for the detachment, and used in the monolayered studies for toxicity and any kind of migration. If you look at the non-adherent cell, that means this is the suspension cell that is particularly float freely in the cell culture plate. That is either the lymphocyte or hybridomas, and they grown in agitation, or so you can grow and agitate the media, and ideal for the bulk production of the antibody and the vaccine. Okay. So based on the shape and appearance, this is the fibroblast, the elongated spindle shape like adherent cell, and in the epithelial-like cell, the growth adherent pact, particularly the polygonal in the shape, and the lymphoblast-like cell, and this is the suspension cell or the non-adherent cell. This is just like a spherical. Okay, so this is the SHSY5Y, particularly the neuroblastoma cell line, and mantle cell lymphoma. This is adherent. This is not suspension. This mantle cell lymphoma or JCO cell is particularly the non-adherent cell or the suspension cell, but the SHSY5Y or neuroblastoma cell line is the adherent cell. Okay. So how to do this kind of cell culture in the laboratory? Just a schematic overview. You first revive the frozen cell, isolate from the tissue or the established cell line. Then you can perform the aseptic condition and then passaging, and ultimately count the cell for the experiment or the cryopreservation. So what is this passaging in that case? So in that case, the cells grow in covering the dish, the confluency, that is called the confluency or confluence in that case. So this is called 20% confluence, this is 50%, 80 and 100. So in that case, at 100% coverage or 90% coverage, they must be passaged. That means the split into the fresh media or new dishes to sustain the healthy proliferation and prevent the overgrowing of the cell. So why you need this passage? First, maintain the cell culture, the good condition without any contamination or over confluency. And how you first check the confluency of the cell regularly, remove the spent media, and then add the fresh media also, and wash with the PBS. And then also, if it is the adherent cell, then you should add the trypsin, and then wash out the trypsin totally, and then you can ultimately perform, or you can get the fresh cell into the culture media. That means the either DMEM or like that, or RTMI 1640, that is specific for the specific cell. So this is the 80% confluency, and this is the 100% confluency. I prefer 90% confluence for passaging the cell. Yes. Hello. Okay. So the cell culture workflow is like that. Hello? Any query? Okay.
It's
So
my
this
request to everyone, please off their video and audio as our eminent speaker is presenting.
And in the next purpose, the cell culture workflow. If you look at the workflow, this is the confluent cell. You can remove the old media and then wash with the previous, and then add trypsin if it is the adherent cell. Otherwise, just agitate the cell, do a centrifuge, and then you can get the pellet. You add the fresh media and then resuspend the cell. Otherwise, you can go with this process. So, what is the cryopreservation? So the excess cell that from the subculturing can be cryopreserved at below 130 degree centigrade, as I told you. So you first, if it is a primary, if it is a secondary or the cell line, you should store the cell into your lab. Okay. So this cryopreservation, why this is necessary? So it can reduce the risk of microbial contamination, reduce the risk of the cross reactivity of the cell, and you should collect this cell in log phase, okay, not in the
Sir,
lag
very
phase
sorry
of
to
the
interrupt you. Sir, very sorry to interrupt you. I would like to request Dr. Bithin Kumar Majhi, please unmute yourself. Please, sir.
Mute yourself.
Your audio is on, so please unmute yourself as our eminent speaker is presenting. It's my earnest request to everyone, please unmute yourselves except our speaker.
May I start?
Yes, sir.
Okay, so sorry for the technical snag. And if you get excess cell or you have lots of cell in the log phase, then that is the 90% viable, and then you just subculturing it by the cryopreservation below minus 130 degree with the DMSO, particularly to prevent the ice damage, that ensuring the long-term viability for the future purpose. So first, it passes the cell, resuspend in the serum containing media, then centrifuge the cell and also aspirate the supernatant, and then resuspend the cell in 10% DMSO with fetal cup serum or FBS, that is the fetal bovine serum. So this is called the cryopreservant media or the freezing media. You can take some freezing vial, that is the cryo vial or Mr. Frosty. This is the name of the cryo vial, and then you can transfer this 1 ml, only the 1 ml of cell. 1 ml means the 10% FBS, 10% DMSO in the fetal bovine serum or fetal calf serum. That particular volume is 1 ml. Okay, but the cell confluence is 90% in that case. So transfer it into first in the minus 80, then minus 130, and then liquid nitrogen. It will ultimately reduce the risk of the damage of the cell. Okay, so if you look at the cell culture, that rapidly the application of the cell culture in the particular purpose that enables the rapid growth and toxicity efficacy, reducing the animal trial for the in vivo experiment. This is the first experiment also, the primary experiment, and high throughput system also applicable in particular specific places for identifying the gaps of the research. You can do the cell culture. So in case of the cellular process, the apoptosis or cell proliferation and cell cycle, you can identify, and also the gene expression level for identify the particular signaling pathway or the target pathway for the disease mechanism purpose. The cell from the patient tissue that is either from the human or any kind of animal for the veterinary purpose. You can treat the cell in the culture media first, and then you can move to in vivo, and after the in vivo you can go to the clinical purpose for the human. And in the case of the cancer research, the primary cell culture in the cancer research uncovering the signaling pathway. You can target the specific signaling pathway for developing any anti-cancerous drug or to reduce the chances of particularly the malignancy in that case. Okay. So the drug screening and toxicity space is very much important nowadays, because the understanding the nanomaterial or other kind of high throughput system is very much important, that integrated the incubators and the plates tracker that particularly the big pharma companies particularly using this technique for screening different kinds of drug by using the cell culture, and also some kind of the 3D culture and the organoid model or organ culture is also nowadays available to overcome any or to reduce particularly any kind of uh, problem or error-free data. Okay, so the understanding of the nanomaterial, that is the nanomaterial immune system, that interaction with the key for the safe as well as the nanomedicine for the regulatory purpose, you can allow the pharma company to screen some thousand of compound and also the valid the in vitro and in vivo model that predict the risk and benefit guideline safety for the therapeutic efficacy. So this is the paper which shows the how this nanoparticle is first treated with in a 6-well plate in a cell culture and then to the in vivo assay for any kind of efficacy test or the protective efficacy or any kind of impact on the cells as well as in the animal model. So before doing any research on the in vivo model, you first should treat anything like any foreign particle into the cell culture model first. That can ultimately maintain our biodiversity also. So if you look at the cell culture technique that particularly can propagate into cell transfer and transplantation and tissue engineering, you first also identify the gene editing repairs that ultimately disease cells in the culture or transplantation or the chimeric antigen receptor, that is the CAR T cell therapy that target the cancer cells specifically, that enhance the cell are re-infused to destroy the tumors, that enabling the precision medicine. And culture of non-mammalian cells that from the plant, particularly the plant tissue culture growth in plant from small sample in the lab, that allowing the mass production of the for particularly the cloning purpose, it also help the propagate rare species and create the genetically modified crop or the GMO in that purpose of the revolutionary agriculture for the conservation purpose also. Okay, so my today's topic is particularly focused on the mammalian cell.
Very sorry to interrupt you, but we are running out of our time, so it will be
just 1 minute. Yes.
Okay.
So in case of the insect culture, we grow in the nutrient-rich media, offering the cost-effective efficacy. In that case, you can look at the SPN or the armyworm in that case. And the bacterial culture, you can either grow with the solid nutrient, particularly for agar small case, and in the large case, you can grow with the nutrient broth for large scale of the culture. Okay. And the viral culture, you need to host, particularly to survive and replicate the host cell, provide the necessary environment for the viral growth. And we will definitely discuss more about the next session or like that. So before doing anything, you first know about the contamination is everywhere, and also the human beings are responsible for that. So it will be better to choose or get precaution from the lab partner. Okay. So in that case, I would like to suggest that it's better to choose a proper lab partner rather than the live partner, because otherwise your experiment will be hampered. Okay, this is just a basic overview of the cell culture in biomedical research. And thank you all for inviting me for sharing my thought. Thank you.
Thanks a lot, sir, for sharing your knowledge and expertise. It was a valuable contribution for us. Let's welcome our next honorable speaker, Dr. Aniruddha Roy. Dr. Aniruddha Roy is an associate professor at the Department of Pharmacy, Birla Institute of Technology and Science, BITS Pilani, Rajasthan, India. He did his B.Pharm and M.Pharm from Jadavpur University, Kolkata, and PhD from the National Institute of Immunology, New Delhi, India. After his PhD, he did his postdoctoral research at the Indian Institute of Technology, Delhi, Ontario Institute of Cancer Research, Toronto, Canada, and the University of British Columbia, Vancouver, Canada. After returning to India, he has been at BITS Pilani. Dr. Roy is an accomplished researcher in the areas of nanomedicine, targeted drug delivery, and regenerative biomaterials. His work bridges the fields of polymer chemistry, cancer biology, immunotherapy, and tissue engineering. He specializes in the development of stimuli-responsive nanoparticles for chemoimmunotherapy, photodynamic therapy, and in designing extracellular matrix-mimetic biomaterials for applications in organoid development and wound healing. Dr. Roy has authored over 44 peer-reviewed publications in high-impact journals with more than 2,500 citations to date. He is the inventor of 4 patented platform technologies, 2 of which are under active consideration for commercialization by FACIT, that is Fight Against Cancer Innovation Trust Canada and Alicor Medical Private Limited India. His translational research experience includes extensive in vitro, ex vivo, and in vivo studies of nanomedicine platforms with an emphasis on tumor microenvironment modulation and regenerative tissue interfaces. He is the lead developer of a thermoresponsive ECM mimetic scaffold for reproducible tumoroid formation. He has secured over rupees 26.8 million or 0.3 million USD in extramural research funding as principal investigator and co-investigator from prestigious agencies including the Department of Biotechnology, Department of Science and Technology SERB, Indian Council of Medical Research, Indo-Canadian Institute and Intus Pharmaceuticals Limited. These projects span advanced cancer nanotherapeutics, theranostic imaging systems, scaffold-based tissue regeneration, drug repurposing, and translational bioengineering devices. Dr. Roy will speak on the topic Animal Models in Biomedical Research. The platform is all yours, sir.
Thank you. I hope I am audible to everyone.
Yes, sir.
Thank you very much for this extensive introduction. So, I hope my screen is visible to you. So, so the topic that I am going to talk about is regarding the animal models in biomedical research and their perspective, the complexities, and I will try to share some of the data, some of the findings that we have based on animal model and use of animal model for different type of biomedical research. So first of all, the perspective. When we talk about the perspective about animal research, we need to keep that in mind that uh, although animal models are extremely important tool for different type of biomedical research purpose, there is no set rule that this is the final destination. And as in science, this the journey becomes much more important, and the animal model that are being used today that may become obsolete in the coming days, and different lot of modification keeps on happening in the model system as well. Likewise, previously there are a lot of different animal models has been used for many different experimentation. Nowadays, those animal models are either obsolete or banned. So, why do we need to use why do we need to use animals? The need to use animal is because of the similarity in the physiology between human and different animals. And to study about the human body and to study about human physiology, different type of animal models play a very, very important role, very significant role. As you can see from here, that there are two different hearts are being projected in this particular slide. and both of them are looking very similar. But if you look, this is actually human heart and this is pig heart. So because of the physiological similarity, any means these animals can be very easily used for this physiological as well as genetic similarity. Now there are many different type of animals are used for experimental purpose, um, and uh, and their percentage actually varies. As you can see from this particular slide, that one particular species is used very commonly among others, and if I ask you to guess which one is that, you will be probably everyone will guess it correctly that most commonly mice is being used for biomedical application because of the small size and easy husbandry and easy upkeeping as well, followed by fish because of the same reason. It is very easy to keep fishes, and means maintenance is quite easy, followed by rats and then birds and other mammals including monkey and reptiles and amphibians. It's a small percentage of experiments. Biomedical research is also done on reptiles and amphibians. However, as reptiles has not significant similarity, and as they are not that easy to maintain as well, so their use is not that prevalent compared to mice. Now, in case of use in the use of these animals in animal research, there is a significant paradox is present. If you ask any researcher that why they want to use these animals for their biomedical research, the most common answer is that because animals have lot of similarity with human. They have physiological similarity, they have genetic similarity, and that's why we are using this animal for in our biomedical experimentation. However, if you ask whether it is morally okay to experiment on animals, then the most common answer is that yes, it is okay to do experiment on animals because animals are not like us. So these two statements make a huge contradiction, and we need to address this contradiction at the very beginning. If we want to do animal experiment, we need to address this contradiction at the very beginning, and we need to figure out that how more ethically we can use animals, because this is an ethical dilemma. This would lead to an ethical dilemma. So So, this paradox can be explained with the help of this particular images, that one same species, they can have different value. The same mouse species, and um, our perception about their utility shapes their fate. Like say for example, if it is a lab mouse, those lab mounts they have protected means they are actually protected research subjects, and they are means they undergo oversights. Many welfare rules are applicable to this animal, whereas there are bad mice as well which are pests, and they can be poisoned, they can be beaten to death. You can do anything to them. But these two animals are the same. They are absolutely, there is no difference. And this mice did not choose to become a pest. It is its means biological. It just end up in the dumpster. And there are feeder ant mice like food for other species, and many such animals are kept as feed for other animals. And there are pet mice as well. They are loved companions, and emotional and ethical values are there. So this lead to this argument that the worth of an animal in research should not depend on convenience or perception, but on our responsibility to use them ethically and meaningfully. Because our our perception should not kind of change their fate. So Jane Goodall, I think almost all of you know about Jane Goodall. So she has a very famous saying that I believe that the more and more people are becoming aware that to use animals thoughtlessly without any anguish or making an effort to find another way diminishes us as human beings. So we should not the actual message is that whenever we are using animals for our own benefit, we should be very careful. We should be careful that we need to use the lowest number if possible, and we should replace it with something else, right? And unnecessarily indiscriminatory use of animals for research is not advisable. So apart from that, we should also make sure that they have adequate food, they have access to fresh water, they need to have a pain and distress free life as much as possible. They should live free of anxiety and fear, and they should able to show, express normal behavior like grooming and hiding what is normally acceptable for that species. And we should do all we can to eliminate or minimize the pain and distress, but we understand that the use of animal in biomedical research, in medical research, is both necessary as well as appropriate. Now, because of that, lot of thought has went into to be based on the ethical use of animals, and these two scientists, Russell and Birch, they proposed a very seminal criteria that we continued using since 1960s. We continue using that, and that principle is known as 3R principle. They talk about 3Rs: replacement, reduction, and refinement. Replacement is substitution, that whether you can substitute the animal experiment with something else, or you can substitute a lower animal species which is not having means that much suffering. Although this is again contradiction, that we cannot decide that whether a monkey suffers more than a mouse. Say for example, that the suffering of a mouse is less than the suffering of a monkey. That is again a very contradictory kind of statement, but we have lot of contradiction in our research methodology. So, and we need to have some logical or some way to do those experiments. So there are some fallacies are still remaining, but the best alternative is to means not using animal altogether. So so we can go towards tissue culture based system, as the previous speaker was talking about, that cell culture based systems are very much useful for the early stage of development. And nowadays we will discuss a little bit on this at the last part of my talk, that we can make more complex cell culture based system that can be used as a replacement for animal experiments. And as well as we can do computer simulations as well, apart rather than using animals at the very early stage of our drug development or any other biomedical research. The second is reduction, that we should be very careful about designing our experiments so that we can use the least number of animals. So we can improve the statistical design so that not high number of animals are required, we can do animal sharing, right? Like one experiment, if it is done, the same animal can be used for some other experiment given some washout time, and we can improve the quality of the animals by genetic modification so that those animals become much more reproducible in result for a specific kind of experiment. So that is another thing that we should keep in mind. And the last one is refinement, that we should design the experiments in such a way to minimize the discomfort to the animal. We should we should try to improve the control of pain. We should try to improve the technical skills of the personnel who are working, so to reduce the pain and sufferings of the animal. And we can try to do less invasive procedures like laparoscopy, endoscopy, magnetic resonance imaging, MRI imaging, micro CT imaging, stuff like that, which does not need sacrifice of the animal, right? So, so these are the major things associated with animal experiment. Now we will discuss a little bit on the complexities of animal use for a specific type of research. As my major area of research is cancer research, so I will focus majorly on the cancer research. What are the complexities of animal models in cancer research? Animal models are used in cancer research at every stage, starting from very early disease identification to the therapy intervention and evaluation of efficacy stage. Everywhere animals are used, and majorly mouse models is used. Um, so, so because of that, um, we need to develop very well properly developments, properly characterized animal model. And because of improperly developed animal model, it has happened before that there are failures in clinical trial has been reported, because the animal model state, in the in vivo stage, the experiment was shown very high efficacy, but at the human clinical trial stage, there means failures was observed. Now, there are quite a few examples are there, like 9-amino camptothecin. It showed strong result in the colon cancer xenograft model. However, it failed in phase 2 trial due to major PKPD difference between mouse and human. Pharmacokinetic and pharmacodynamic, PK and PT, they are not the same between human and mouse and human. So whatever data you are obtaining from mouse cannot be directly interpolated for humans. And similar kind of failures has been reported for other kind of therapy as well, indicating that proper development of an animal model is very much necessary. So this kind of failure not only makes all the animals that are used in this study, just they are just completely wasted, as well as in the clinical trial. Clinical trial is a very expensive process, and that is also completely wasted. That much money got completely wasted. So for development of proper cancer model, there are means from the historical perspective, many different kind of development has been reported. The first generation of tumor model is dependent on grafted tumor cells, where you take tumor cell from either a patient or from a cell line, and those patient derived tumors or the cell line tumor is grafted in a animal, most commonly mouse, and they allow the growth of the tumor. So second generation is transgenic model, that that is when you design an animal in such a way that a specific gene will turn on a specific tissue at a specific time point, and that turning on of that oncogene would lead to development of a tumor in that specific tissue. And then you can study the tumor development and tumor progression and efficacy of different type of treatment. So that was the second generation tumor model. Third generation tumor model is advanced transgenic model, which when you can specifically, spatially and temporally, that means space and time, which tissue at what time, you can control the tumor formation by turning on or turning off of a specific gene, turning off of a oncogenic gene, or turning off of an anti-oncogenic gene. The fourth generation includes spontaneous tumor models mimicking tumor evolution and patient-derived xenograft. The fifth generation is advanced third and fourth generation models mimicking metastatic progression and high throughput tumor models in target validation state. Now, the tumor model development in animals are done through many different routes. The tumor can be implanted through tail vein inoculation. Tail vein inoculation will lead to metastatic kind of signature, because the tumor cells are going into systemic circulation directly. That would lead to kind of metastatic kind of nodule formation, or the tumor cell can be transplanted through subcutaneous inoculation. They can also be transplanted through orthotopic inoculation, where the tumor cell is implanted to the tissue where the tumor is supposed to grow. So, for example, you are trying to develop a breast cancer model. So that breast cancer, those cancer cell can be transplanted through subcutaneous injection. That would lead to a subcutaneous breast cancer model. At the same time, those breast cancer tumor cell can be implanted in the breast fat pad as well, and that would be an orthotopic tumor model, because that would lead to tumor growth in the breast fat pad where the actual tumor grows. The same thing can be done for lung cancer, for colon cancer, for brain cancer, for many different type of solid tumor. The, the tumor cell can be implanted at that specific tissue, and the tumor growth can be analyzed. So those are known as orthotopic inoculation. Retroorbital inoculation can also be done,
You're
which
ready to
is
join, no?
mainly,
This is
which is
not
very
ended
similar
second.
to as that
Okay,
of
okay.
tail vein inoculation.
So when you're ready,
Also,
you can join,
that also
because
replicates
I'm not sure
that
when this will end,
metastatic
but
tumor.
half. Yeah,
Um,
yeah. Okay,
Tumor
thanks a
bearing
lot.
mice can be
Yeah,
of
sure,
two
sure,
different
sure. Okay.
types. One is syngenic model. Syngenic model means when you are using a mouse specific cancer cell line, the cancer cell line which can grow in that specific mouse. Say for example, a very common example is 41. 41 is a breast cancer cell line which grow in bulb C mice, which is syngenic to bulb C, right? They are HLA matched. Those 41 tumor cell lines, they are isolated from 40 mice only, Balbsi mice only. So those 41 tumors, so they can grow very easily in Balbsi mice, because they are identical, genetically identical. So that kind of model is known as syngenic model. Um, and apart from syngenic model, so the advantages associated with syngenic model is that they are well characterized cell lines, those cell lines that are used. They are immunocompetent host. You are injecting, you are growing the tumor in a immunocompetent animal. So the normal physiology and the involvement of the immune cells can all be studied in that. They are quite reproducible tumor. They produce tumors quite reproducibly. not much variation is observed. Almost very commonly, uh, they produce reproducible tumor and they are low cost. However, the disadvantage include their poor representation of human disease because they are all animal origin. The cell line that you are using is of animal origin, and although they are similar to human, but their expression pattern is not exactly same to human. So because of small variation, you will not be able to replicate exactly human disease. And there are lack of target molecule homology between species. So if you are using a immunotherapeutic molecule like antibody or that kind of biologics for evaluation, these syngenic models are not the best one, because the antibody that you have developed to target human cell, they may not target mouse cell with that same kind of activity. So, so these are good for small molecule based therapy, chemotherapy. The next one is xenogenic model, where you isolate the tumor cell from human, and the xenotransplantation you are doing. Xenotransplantation has a significant disadvantage that they will not grow in an immunocompetent mouse. If you transplant human cell in an immunocompetent mice, the the animal will immunologically reject the cell line, and because of HLA not matching, right? Because the histocompatibility is not there, so the immune system will be able to recognize those transplanted tumor cells as foreign cell, and they will eliminate them quite easily. So we need to use immune suppressed mice like nude mice or skid mice for growing this kind of xenogenic model. Now, the xenogenic model, the advantage include they are well characterized cells can be used. They are relatively simple to implant. Expression of human homologous target is there. So if you are evaluating the efficacy of any immunotherapeutics, that can be done using this kind of model. Homogeneity in tumor, and they are relatively reproducible as well. So, however, the disadvantages or the means negative points of xenogenic model is that as you are using an immunosuppressed host, the influence of immune cell on the tumor growth or your efficacy you cannot be able to evaluate. Murine peritumoral milieu is there. It is not like human milieu, so the tumoral microenvironment that is being created, that will be of mouse origin. Although the tumor cells are of human origin, but in a solid tumor, apart from the tumor cell, there are a lot of other cells also get involved, like fibroblast, macrophage, neutrophil, many different type of cells, they get involved in that tumor microenvironment, and those non-tumor cells will be of mouse origin, which would lead to differences. There will be an immunosuppressed microenvironment of tumor. You will not be able to evaluate the efficacy of immunotherapeutics. Different human tumors, histology more expensive, and it is significantly more expensive because of the involvement of immunosuppressed mice, and they themselves are much more costly, their husbandry is much more complicated, and so on and so forth. And another thing is that that the tumor morphology can be reproduced in xenograft. So this is the human patient morphology for triple negative breast cancer, and this is the xenograft morphology. As you can see, they are quite similar, right? They are quite similar to each other, and the same thing can be seen with HER2 positive tumor cells. as the patient morphology and the xenograft morphology is very similar, right? So, however, this morphology is generally transiently present, and gradually that this the tumor microenvironment is replaced by mouse trauma. The tumor microenvironment that is there, as we have discussed, that presence of other type of cells are there, those cells will gradually will be replaced by the human cell will be replaced by mouse cell. So based on that, so these are all means preliminary type of immune cell, preliminary type of animal model. Advanced model, as we have discussed, that advanced model include transgenic mouse. In that you can actually control the expression of the tumor production by turning on or turning off of specific gene. So you need to develop an animal, a genetically modified animal, and that in that genetically modified animal, specific gene expression will be able, you will be able to control the expression of specific gene. Genetically modified animals can be developed in many different ways. The one easy way of using this retroviral route, where you take a donor female mice, you take out the embryo, and you treat the embryo with retrovirus carrying some transgene, and those transgenic means after modifying the mouse embryo with the transgene, you implant it in the mouse, and then you try to figure out that which of the offspring is having that transgene. So this is one of the strategy for making genetically modified mouse. The second strategy is using the donor female mice, and then you micro inject the that specific genetically modified DNA in the fertilized oocyte, and then you grow it, and then you transplant it into the mouse, and in the litter, in the offspring, you will get the genetically modified means genetically modified offspring you will get. Another strategy is to take embryonic stem cell, and then treating those embryonic stem cell with DNA with that transgene, and then the DNA can be introduced into the embryonic stem cell, and that then injected into the mouse embryo. Those embryonic stem cells can be injected into the mouse embryo, and then among the litters, there will be litters which are having those transgene in their um, in their germ cell, and those animals which are having those transgene in their germ cell, that means in the sperm and ova cell, they can transmit, they can keep on passing on those genes into their next generation. So you can make a transgenic line by using that strategy. So when you are able to make such kind of genetically modified mouse, what you can do, you can selectively turn on and or turn off the expression of specific genes in that animal, right? That specific turn on and turn off you can do. However, the transgenic animal, although they are very good model, however, they have limited availability, they are quite expensive. You need to have a lot of experience to work with them. There are variations in tumor growth, and this and that is there. Apart from that, scientists have also developed humanized mice by injecting human bone marrow in an irradiated mouse. So you take a normal mouse that is being irradiated with gamma ray to kill all the bone marrow cells. So the mouse become completely immunosuppressant. All the immune cells have died. Now human bone marrow is transplanted in that mouse, so that the immune system of that mouse is now replaced with human immune system. And then, so this mouse will be having a mouse but with human immune system. So you can implant human cell in that mouse to grow human tumor, and it is an immunocompetent mouse altogether, right? It is carrying human immune system. So using this humanized mice, you can check out the efficacy of any this uh, this antibody based therapeutics, right? Immunotherapeutics can be evaluated using this humanized mice. So these are the advantage and disadvantage of different mouse cancer models. Now, at the last part, we will discuss, another 5 minutes I will take to discuss about some of our own findings in using mouse model for cancer. This is the first one. So this is a very simple experimentation that you grow tumors in the mouse, subcutaneous tumor implantation. You give different type of treatment and you check the tumor size, right? The tumor size can be evaluated as well as the tumor weight. At the end of the experiment, you can sacrifice the animal, isolate the tumor, and you can check out the tumor weight, and you can try to figure out which of the therapeutics that you are using is having the lowest tumor growth and lightest tumor weight, the smallest tumor weight. So apart from that, what you can do, if you are trying to figure out the efficacy of an immunotherapeutics, then you can isolate the tumor infiltrating immune cell and you can do FACS analysis, right? And in that FACS analysis, you can try to figure out the amount of tumor infiltrated cells, like here CD14, CD11C, CD4, CD8. The population of this cell inside the tumor has been evaluated. CD14 is a marker of macrophage, CD11C is a marker for dendritic cell, CD4, CD8 is marker for T cell, and so on and so forth. So there, how much tumor infiltration is happening for different type of therapeutics, that also you can study. The second one is very similar to the previous study only. However, the 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 one more thing is here is that you can check out double marker, right? CD14, CD40. CD14 is the marker for macrophage and CD40 is the activation marker. So among the CD40 cells, CD14 cell macrophages, how many are the activated macrophage, right? CD40, CD80, CD83, these are all activation markers. So you can check out the activation of those immune cells in the tumor model, in the tumor animal tumor model. Now, for all this kind of experiment, you need to use an immunocompetent mice. Immune suppressed mice, you will not be able to evaluate. This the next one is discussed about that different type of tumor cells have been taken and the same therapy has been given. Very interestingly, you can find that the same treatment is not as effective against all type of tumor. This is MDMB231, a human breast cancer cell, PC3 mouse breast cancer, human prostate cancer cell, LL2 is lung cancer, EMT6 is breast cancer again, B16FT is melanoma, right? So the same therapy is not equally effective against all type of tumor. So that you can evaluate at an early evaluation stage using animal model, that for which kind of tumor your therapy will be the most effective. Very early animal model study you can do. This is another very interesting study using cancer targeted nanoparticle, you can evaluate in an animal model that how that your therapeutics is going to the tumor. As you can see here, these are this is tumor, this is liver, and this is spleen, and the red color indicates the accumulation of that particular therapeutics. And these are three different type of nanoparticles. This is 20 nanometer nanoparticle, this is 30 nanometer, this is 120 nanometer. So very interesting thing has been observed is that 20 nanometer nanoparticle, their tumor accumulation is significantly more. And 120 nanometer nanoparticle, they are getting accumulated in the liver. So this kind of early research can be done in animal model, using animal model quite easily if you can design the experiment properly. Also, intratumoral distribution of the therapeutics, that can also be done using animal model, that you take the tumor out after the experiment is over and after sacrificing the animal, and you can evaluate inside the tumor how your therapeutics is getting distributed, right? So that kind of experiment can also be done. This is kind of extension of that study. Here, a similar kind of nanoformulation was developed, and it was checked out the ability of that formulation to go to the lung metastatic tumor, and it was observed that in the tumor bearing lung, there is significant accumulation of the therapeutics. Non-tumor bearing lung, there is very low accumulation of the tumor. The last data, this is again a very interesting model. Here, the tumor cells themselves are carrying, this is actually a genetically modified tumor cell. The tumor cells are luciferin tagged, and so because they are luciferin tagged, they give out this luciferin signal, and using an in vivo imaging system, you can monitor the growth of the actual tumor from outside without sacrificing the animal, because the more the tumor will grow, say for example this is a control animal, as you can see the fluorescence signal has constantly increased from day 0 to day 21. There is a significant increase in the fluorescence signal, whereas this is a treated animal, and as you can see that there is not significant increase in the fluorescence signal, because that is signifying the efficacy of the treatment. So so this is in this kind of animal model, you can very easily evaluate the efficacy of the tumor, efficacy of a therapy using a proper animal model. Right now, as we have discussed in the very early stage part of our presentation, that apart from using animals, we should also be careful and try to replace animal model. So for the replacement of the animal model in our lab, we have developed a very specialized kind of 3D tumor system. I'm not showing any data as of here now, because it is we have yet to communicate the paper. We got really excellent data, but the theory I would like to discuss over here, that generally what happens, that when we do 2D cell culture, these 2D cell cultures are not actually replicate the actual human tumor. Human tumors are a 3D kind of 3D cell growth, right? Now to replicate that one, many scientists have developed 3D tumoroid. These 3D tumoroids are nothing but cell aggregate. All the tumor cells, they come together and make a cell aggregate in 3D. They are no longer growing in the planar kind of in a petri plate. They make an aggregate. However, those aggregate lacks any extracellular matrix. If you look into actual tumor, you will find those actual tumor have extracellular matrix which is holding that cell together. Now, this kind of normal 3D tumoroids, they have the disadvantage that their core become necrotic very easily, right? Because they do not, because they are densely crowded cell, and they do not get the nutrient very easily, so they become necrotic. So we have developed this kind of an ECM mimetic scaffold based tumoroid. So as you can see, that this kind of scaffold based tumoroid, lot of people have tried to develop, and they generally use matrix like matrigel. So this is a same image, scanning electron microscopic image of matrigel. As you can see, the matrigel, this bar indicates 1 micrometer. So the matrigel is not very highly porous. If this is 1 micrometer, this is a tumor cell. Tumor cell diameter is generally around 20 micrometer. So you can see, you can understand that if a tumor cell you place, you put it on top of matrigel, they will grow on the surface only. They will never go inside the matrix, right? Because the porosity is very, very low. However, you need to have some matrix where the cell can grow inside. So we developed such kind of a matrix. I am not disclosing the component here, because we are yet to publish that paper. So this is having a pore size like this. The pore size is more than like 200 micrometer. So tumor cells can very easily grow inside that. So this is a same image of tumor cells growing in that matrix. So this is a tumoroid, a 3D tumoroid formed using that matrix, and we can do many kind of thing using that matrix. First of all, what we can do, we can use this kind of 3D based model for high throughput drug screening using tumor cell line, as well as we can take patient derived tumor cell and we can figure out biomarker expression in that patient derived tumors for to predict drug action, as well as we can grow the patient derived tumor cells in that kind of a 3D matrix, and we can check out differential drug response, right, for personalized therapy. This kind of matrix is very, very valuable, because maintenance of primary cell means surviving of primary cells in cell culture is very difficult. We generally do not able to do that. So in conclusion, what we can say, by replacement, reduction and refinement, it remains the ethical cornerstone for animal experimentation, and we need to do human research based on that. Careful selection and validation of animal models is very much essential to do a proper animal experiment, so that uh, we can predict all the things. And our data using model development, experimental design, and replacement of animal model, as we have shown, that can add scientific value and as well as societal impact for advanced biomedical research. So with that, I thank you very much again for your invitation, kind invitation, and listening to the talk. I will be very happy if you have any question. I would be very happy to answer that.
Sir, we have the Q&A session at the end of the three speakers lecture.
Okay, thank you.
Thank you, sir. Thanks a lot. Your talk has provided us with a new perspective on animal models in biomedical research, and I, along with many others, found it incredibly beneficial. It's time to welcome our final most speaker, Dr. Vinay Chituri. Dr. Vinay Chituri is an associate of Physiology, All India Institute of Medical Sciences, AIIMS, Rajkot, Gujarat, India. He did his MBBS from the Jawaharlal Institute of Postgraduate Medical Education and Research, Puducherry, and MD in Physiology from AIIMS, New Delhi, India. He worked as a senior resident at AIIMS, New Delhi from 2017 to 2020, after which he joined AIIMS Rajkot. His research interests include clinical neurophysiology, quantitative electroencephalography, and digital signal processing. Dr. Chituri will speak on the topic ECG and its application in clinical research. The platform is all yours, sir.
Thank you so much, ma'am, for the kind introduction. I'll start presenting my presentation. Okay. I hope the slides are visible.
Yes, it's visible.
Ah, so very good evening, everyone. So I'll be presenting a topic which I believe is amongst the easiest research protocols to run because of its low barrier to entry, right? Electrocardiography and electrocardiography based research protocols and research applications. So I'll start off with uh, the modern style of YouTube video where they're all about the highlights. So uh, with the assumption that all of us have a fundamental undergraduate level understanding of ECG, I'll highlight some of the key resources which are useful for learning of ECG and some of the research tools available to us, right? So typically, whenever we teach undergrads ECG, I found this book to be very good at explaining the conceptual foundations of ECG, the ECG Made Easy by John Hampton, right? And then once you know the basics of ECG, if you want to train yourself in the interpretation of clinical ECG, I found the Harvard Maven database. I'll be showing this later. But the Harvard Maven Clinical ECG database is a wonderful tool for people who wish to train themselves on clinical ECG reporting. Now, in addition to that, if you're looking for large data sets, right? So there's a machine learning challenge out there which makes an open source data set, and people are tasked with designing classifier protocols to accurately classify the clinical ECGs. So there are data sets out there, and these data sets can be used for validation of your own AI ML workflows. So these are some wonderful resources. Now, assuming people already know what heart rate variability is, heart rate variability is this ECG based RR variability analysis. So, typically, it comes with inbuilt hardware with inbuilt software which runs HRV. But in case you do not have those inbuilt hardware, you can always consider using an open source toolbox to learn the mathematical operations of HRV. So, QBOS and PhysioConnect have some very good toolboxes, right? Then you can also code your own toolbox. Then similarly, if you're looking for large data, HRV, prior recorded data for AI ML applications, you can get them from my alma mater, AIIMS Delhi Department of Physiology, Dr. Deepak, my old HOD, has a database lasting over 30 years, and then there's some global databases too. So those are the highlights. Now I'll start going into more conventional. What are the fundamentals of ECG? So electrocardiography, electrical record of the cardiac activity. Now typically what leads to this electrical activity? So if you'll see, there's a specific sequence of depolarization of the heart. So it's showing a ventricular septum depolarizes first. So at any given time, some parts of the heart are depolarized, the other parts are RMP at resting state. So there's an electrical potential difference between different parts of the heart. This leads to a configuration called a cardiac dipole, right? Cardiac dipoles produce electric fields. Electric fields lead to potential differences at different places, let's say point A and point B. And these potential differences can be measured as a way of studying the cardiac electric field. So ECG at its simplest is a potential difference between 2 points in the cardiac electric field, right? So what leads to the typical ECG as you see. So this is a sequence of depolarization starting from the SA node, spreading across the atrium, connecting to the ventricles, and then depolarization follows the sequence. So that specific sequence gives us the waveform of ECG as we know it, right? So P waves are atrial depolarization, QRS is ventricular depolarization, T wave is a sequence due to ventricular depolarization. So as and when the heart rate, as and when the heart produces rhythmic cycles as per the heart rate, we see the typical waveforms associated with ECG, right? The P wave, QRS complex, E wave. right? So typically lead 2 has a vector of 60 degrees aligning with the cardiac vector for QRS complex in most cases. So that's why lead 2 is typically preferred to measure if you have to record a single channel ECG, right? Now typically in a clinical setting, ECG would look something like this. You have a multi-channel ECG device. You place multiple electrodes, typically 4 on the lens, 6 on the chest, and you record a combination of leads from these electrodes called a 12 lead ECG. Looks something like this. So why do we need 12 leads? So typically each lead has a separate vector system. It's like a camera angle. So like in a cricket match, if you need multiple camera angles to get your DRS system right, ECG vectors also give us different camera angles to be capturing the electrical activity of the heart. Certain cardiac vectors are better represented in some leads than others. So a 12 lead ECG represents a global coverage of the electric field both in the coronal plane and horizontal plane. Now typically a 12 lead ECG would look like this: bipolar limb leads, augmented unipolar limb leads, and 6 chest leads. Then you also have a 10-second rhythm shift. So typically in a clinical setting, this is what a 12 lead ECG would look like. Right? Now how do we go about interpreting this ECG? So typically this is a systematic sequence. We look based on the ECG. This is the rate, rhythm, axis or the QRS axis, QRS complex, intervals and durations of key intervals, such that would be PR interval, um, uh, QRS complex duration, QT interval, and RR interval. Then we look for individual waveform abnormalities in the P wave, PR segment, QRS complex, ST segment, and T wave. Then you look for overall interpretation. So that's how we go about it. So it's a systematic process, and based on this, you can get a clinical diagnosis if you follow the process. Now, ECG primarily and first and foremost is a clinical investigation. We see this of regular use in cardiology patients. So typically, the wisdom as it goes in medical science, anyone coming to you with chest pain or non-specific abdominal pain, always we get an ECG to rule out myocardial infarctions. So typically, mostly we see ECGs recorded in a setting of myocardial ischemia or in worst cases, myocardial infarction. So typically we get repolarization currents, abnormal repolarization currents, abnormal injury currents of systole and diastole. So this gives rise to a typical finding called ST segment elevation in myocardial infarction. So this is a diagnostic tool to identify myocardial infarction, one of the first line investigations. Then secondly, if there are any disorders of rhythm or conduction, so it could be conduction blocks, it could be atrial supraventricular arrhythmias, it could be ventricular arrhythmias. So this ECG is an ECG of a ventricular fibrillation, a medical emergency. So disorders of rhythm and conduction can also be diagnosed by ECG. So for myocardial ischemia, the acute coronary syndromes and arrhythmias and conduction disorders of the heart,
প্রনিশা বলছি
ECG is a first line diagnostic
তো বেরিয়ে
investigation.
গেছে
Now, other than this,
নেই
there are
এখন
some other cardiac
ও
disorders
চেষ্টা
like
করবো
cardiac
পরে ঢুকতে
hypertrophy,
মোবাইলেও
hypokalemia,
বাইরে
metabolic
গেছে আর কি
abnormalities,
মানে
in which
আউটসাইড মানে বিল্ডিংয়ে
ECG
নেই
can be a second
তো
line
যদি
investigation
না
for the
থাকে তুমি একটু
overall
চেক করে
evaluation
নিও
and management of the
কোয়েশ্চেন আর
patient.
কিউএন এর সময় যদি
Then the
না থাকে
primary
তাহলে একটু
investigations will
বলে
be
দিও
something else.
যে
Now, also in an
যদি
ICU
কোয়েশ্চেন
setting,
থাকে
we
তাহলে
want to monitor the
ইমেলে
vitals
দিয়ে
of the
দেবে
subject. Heart
ঠিক
rate is
আছে
a very important vital to track cardiac function
হ্যাঁ হ্যাঁ
and cardiac output
ইমেলটা
indirectly.
দিয়ে দেবে
So
বলে দেবে
BP
যে
and ECG
ডক্টর
are
অনুরোধন
routinely
থাকতে
measured
পারছে না
in cardiac
কাজে তো
vectors
পরে
in
ওখানে
emergency and
পাঠিয়ে দিতে ঠিক
ICU
আছে
settings. So
ওই
these are the primary clinical uses
হ্যাঁ
of ECG.
had to leave
Right?
he had to
Now,
leave due
based
to
on this,
urgent engagement
based on clinical
কিন্তু
ECG,
তার আগে এবার দেখে নেবে ও আছে কি নেই
there's an
একবার দেখে
extended
নেবে
version of
ঠিক
clinical
আছে
ECG which is not routinely done, but
ওগুলো
which
আমার
gives you
আমার
a
ইমেলে দিলে
quantitative
ওটা
parameter
আমার
which you can measure.
স্যাপের ইমেল
So I
ওটাতে দিলে
talked
আমি
about cardiac
অনিরুদ্ধকে
dipoles
পাঠিয়ে দেবো
earlier.
কোয়েশ্চেন্সগুলো
So a cardiac dipole is basically between
হ্যাঁ হ্যাঁ
depolarized
হ্যাঁ
and
স্যাপ ডট
resting
রিসার্চ
state
ডট
muscle.
ওইটা
So at any given
হ্যাঁ
point
হ্যাঁ
of time, depending on the
তো
pattern of
ওটাতে
execution of the heart, you have a dipole of a
হ্যাঁ
specific
ঠিকঠাক আছে
magnitude and an
ঠিকঠাক আছে
orientation.
ভেরি
So
গুড
as and when the cardiac cycle progresses, the
হ্যাঁ
dipole changes its orientation, the
না
dipole
না
changes
না
in
না জয়েন
magnitude.
করেনি
So if you
না
plot
করেনি
this cardiac dipole in 3 dimensions, right? You
মনে
get
হয়
you get a
মনে
graph
হয়
called a
করবে না
vector
হয়তো যদি
cardiogram,
করে
ECG.
তাহলে
Right?
বললো
So this is a
মানে
simple
বলবে
calculation based on
আমার সঙ্গে
the
কথা
recordings
হয়েছে
of
সামনের সঙ্গে
at least 3
তো
leads
ও
as
আর কি
recorded in
হয়তো
typical
জয়েন করতে
quality
পারবে
ECG.
না আর কি
The software
আমি
tools
বলেছি
which
যে
do
ঠিক
this
আছে অসুবিধা
automatically.
নেই
So the connector
বলে
cardiogram
দিয়েছি। তো
is an expansion of clinical
ওটার একটা
ECG,
এনগেজমেন্ট
which
রয়েছে আর
is
কি, ওই
used in
জন্যে
typically
তুমি
cardiac
একবার দেখতে থেকো,
units as
যদি জয়েন
a
করো। আর
marker
আমি
of
যদি
cardiac
দেখতে পাই,
function.
আমি তো অবশ্যই,
So
আমি
vector
অ্যাডমিট
cardiogram,
করছি তো
we get
সবাইকে, আমি
vectors for
দেখতেই
the P
পাবো।
wave, we get
তো আমি
vectors for
তোমাকে
the
ফোন
T
করে
wave,
বলে দেবো
we get
যদি
vectors
এসে
for the
থাকে।
QRS
ঠিক আছে?
complex. So
আচ্ছা
BCG
আচ্ছা,
is a
রাখছি
mathematical
তাহলে।
calculation run on a typical ECG recording, a typical multi-lead ECG recording. Right? So we are getting you're mapping the vector in 3D space, and we can easily do it. Now AI and ML is all over the place nowadays, right? So clinical diagnostics are encroached upon heavily by AI applications where people are looking to develop classifiers which can detect clinical syndromes based on either single source of data or multiple sources of data. And there are whole machine learning challenges out there where people upload clinical labeled data of clinical ECGs, and researchers and students are encouraged to develop machine learning and deep learning based algorithms to classify these abnormal ECGs and make a prediction. Right? So this is a well-known machine learning challenge. So anyone who wishes to work in these kind of settings, AI machine learning. So as they like to say, in AI and machine learning, the biggest challenge is data collection and data pre-processing. So there are people who have collected this data and made it available for people to use. So now coming to a clinical ECG background. So if you're an entry level undergraduate or someone who wants to brush up their ECGs, I would suggest this sequence. Oh, the fundamentals must be gotten right every time. So ECG Made Easy by John Hampton. This is the book to read for a fundamental introduction to ECG. Then once that is done, right, you can always challenge yourself with with your ability to judge. Wait, sorry. Sorry about that. You can always test yourself with clinical ECG database. So I use this database extensively to teach my undergraduates. So there are 500 ECGs which you can use. So for example, there are clinical syndromes and there are well-labeled clinical ECGs where a good ECG is presented, and we teach our undergrads on this. So there are 500 ECGs on this, some of them easy, some of them difficult, but overall a wonderful resource to be learning clinical ECG. So someone who wishes to train themselves in the interpretation of clinical and diagnostic ECG, Howard Maven is a wonderful database to begin with once you assert yourself on the fundamentals. Now, similarly, if you're looking for a large ECG data set for AI and ML trainings, so PhysioConnect is a resource which you can be using, right? So these are all these ECG-based databases which you can see. So, for example, this is an arrhythmia database of around 10,000 ECGs. So, this is open source data which you can download and use this for your ML. So, a nice 2.3 GB of ECG. So, Kaggle and PhysioConnect. So, these are some wonderful open source large data set repositories which are available for everyone who is looking for AI based research on pre recorded data set. Now, a secondary clinical application of ECG is in the domains of cardiac autonomic function testing. Right, so we typically do this in the setting of patients who come to us with orthostatic hypotension, typically syncopal attack, loss of consciousness, lightheadedness, giddiness, or taking the standing position from a supine position. So many causes for orthostatic hypotension. One of the causes is autonomic dysfunction. We see this in long-standing diabetes typically. So there are ECG and BP based tests. So they are designed as bedside tests. At the least, we can do this in a field setting with just a typical ECG device and a simple sphygmomanometer. So they are designed to assess the cardiovascular, sympathetic and parasympathetic reactive stressor. And cardioautonomic dysfunction is a comorbidity typically assessed in long-standing diseases of many chronic long-standing diseases like diabetes, renal diseases. So you can use these 5 tests: postural challenge, delta BP change of posture from lying to standing or in a tilt table, deep breathing, It's a small 1 minute test of slow breathing, and you look for heart rate and RR interval changes. Valsalva maneuver, a specific maneuver presented in the upper airways, designed to act as to reduce the venous return and act as a stressor of the heart. Then there are some BT based measures like isometric handicap and cold pressure. So this is a battery of tests called the Ewing battery to assess comprehensive cardiac autonomic function. It's a simple protocol. This entire protocol, if you club it with heart rate variability, takes less than an hour to execute. So typically we do it in around 45 to 50 minutes. So it's a simple protocol called the Ewing's battery. The Ewing's battery can be used as an assessor of cardio-autonomic function. Now, then coming to one of the most common research tools of ECG, heart rate variability or HRV, right? So, a brief primer on what HRV is. So, this is a typical single channel ECG signal, right? So, you can see multiple cycles of ECG. So the time between one QRS complex and next cycle's QRS complex is called the RR interval. So if we go about measuring the RR intervals of each cycle and all subsequent cycles, right? So if you have multiple, let's say we have some 30 cycles, we get 30 RR intervals. Now one thing to notice, RR intervals tend to vary a bit from beat to beat, right? This is routine heart rate variability. This is because of the integrated cardiovascular respiratory control systems. So things like respiration, BP changes, sympathetic and parasympathetic tone can change your heart rate and thereby affect heart rate variability. So if I take these RR intervals and plot them interval cycle by cycle, we get something called an RR tachogram. So first cycle, this was RR interval. So we had 400 odd cycles. The RR interval is changing in a specific way over time. So this is called an RR tachogram. Heart rate variability is basically a study of variability in RR intervals, right? So to typically get a graph like this, easiest way is to get a digital ECG, and if you have a digital ECG, there are mathematical algorithms and toolboxes or inbuilt custom solutions which allow you to quantify RR variability and all the subsequent HRV analysis based on RR variability, right? So just a primer on some of the quantitative parameters which we can measure from RR intervariability or HRV measures. So there are time domain measures which basically are a statistical quantification of how much variation is there in the RR interval as it changes with time across multiple cycles. So these are well established parameters, well documented. Anyone working in HRV would be using this. SACE-ER, standard deviation of RR intervals, is considered to be a very good correlate of cardio-autonomic tone. But these are all parameters which you can be using and reporting from a single ECG channel recording, right? So you get time domain parameters. Now similarly, if we see the RR interval, there is a certain pattern of oscillation. So I see a higher frequency oscillation superposed on top of a lower frequency oscillations. So to quantify this pattern of oscillation across multiple frequencies, we do something called a frequency domain analysis. So there's a tool, either a periodogram or a fast Fourier transform, mathematical algorithm, typically run by software automatically, would convert this into a frequency domain plot. So in a frequency domain plot, you get different frequencies, and the amount of oscillation in this frequency is typically quantified as a spectral power, right? So frequency domain measures can also be quantified in terms of the amount of strength of oscillation. So we have numerical parameters like the power, spectral power present in different frequency bands of ECG, power ratios. Now other than that, there is patterns of variability. So if you plot RR interval against the next RR interval in the RR tachogram variability, we get this kind of scatter plot. So there's an elliptical pattern fitting to this, and based on this dimensions of the ellipse, we get something called non-linear measures like ST1, ST2, and triangle vertex related non-linear measure. So based on this, you can use HRV. So typically people do this in a 5-minute recording, and 5 minutes you can run time domain analysis, frequency domain analysis, and non-linear analysis. So what is the typical methodology of a HRV recording? So first and foremost, you need a source of digital ECG. pen and paper ECG might do the job, but then you'll have to measure RR intervals manually, and that's very inefficient. We need a source of digital ECG, comes in many forms, shapes, and sizes. And as of nowadays, there are wearable devices which allow you to give you either ECG or a PPG signal, can be used interchangeably in most cases of HIV analysis. Right now, how long does this ECG needs to be? So typically, most protocols do a short-term HIV protocol, 5 minutes, but you might see yourself doing longer-term recordings lasting. So HIV protocols are run from ultra short-term 1 minute to ultra long-term 24-hour recordings. So typically, you choose hardware as appropriate for the recording you're trying to do. So get a good duration of digital ECG, extract the error intervals. It's a simple mathematical operation. You have to do this manually. The softwares and algorithms which do that, right? Then we can run the standardized HRV algorithms of time domain, frequency domain, and non-linear parameters to compute HRV parameters. And these HRV parameters are quantifiable outcome parameters which can be of use in your research, right? So typically you get a whole bunch of parameters like this. Now when and how would you use these parameters comes from your study design, right? So little preview of how can you go about recording ECGs. So if you are recording ECGs as we would do in academia studies, a portable device is the way to go, something called a Holter monitor. The custom built HRV monitors and devices, right? It's typically connected to a laptop, slightly less portable, but portable enough to be taken out into the fields and doing your studies. Then the modern revolution is wearable ECG and PPG devices. So there are a whole bunch of FDA approved wearables, Apple Watch, Samsung watches, which automatically measure BP in the recordings. And in today's HRV recordings, if you're doing long-term HRV recordings, I would recommend using a wearable device. That's fairly easy to procure and run. Right? Now, once you have recorded a digital ECG, how would you go about analyzing it? Now, the graphs I showed you were used in the inbuilt software which we recorded the ECG on. So, the software analysts things. But if you do not have the software, you can use tools like QBS HRV, right? Right? So, it's an open source toolbox we can run. Upload your ECGs in a standardized text or a CSV format, and the HRV analysis is run for you in different ways, right? Similarly, PhysioConnect, a wonderful database for both toolboxes and open source data, has cardiovascular signal toolbox. Now, if you find yourself doing batch analysis on large data sets, you won't reasonably clicking around on these toolboxes, running the algorithm manually on each individual data file. So in those cases, you would need an efficient solution. In that case, I would suggest generating a script on a scripting language or a programming language like MATLAB, Python, and R, and batch run your ECG analysis automatically. Makes it very easy to integrate your data storage, data processing, and subsequent data statistics and plotting. So, in case you're working with large data, programming is the way to go. And if you're looking for HIV databases to be running your research on, Ames Delhi has a database lasting over 30 years, and Physiological Title is the wonderful source you can be using. Right? Now, once you have recorded HRV, what are the outcomes you are looking to report on? So, HRV first and foremost in the 1980s was developed as a prognosticable biomarker for cardiovascular risk. So, typically, patients of myocardial infarction and diseased hearts were found to have severely reduced HRV. And even today, we consider reduced HRV as a marker of reduced cardiovascular tone and a cardiovascular risk marker. Now, HRV is used extensively as a psychophysical study to quantify things like arousal, emotional responses, and stress. And you can also use HRV in biofeedback-based applications. There are actually softwares which integrate with your wearable device and program a biofeedback device. So HRV is compared to some of our previous speakers who are using very excellent research tools, but these research tools I would say have a fairly high barrier to entry. ECG and HRV have a very low barrier to entry. So typically in our MBBS colleges, we have ICMR STS undergrad medical students. Sometimes we even see high school students also doing HIV studies. So just to give you a small preview on the things we do in HIV, so I thought I would just pull up some of the things we have reported. So we have a study on yoga, right? Then we have some other studies. So I would say around half my publications have HIV-based recordings, right? So it's a wonderful physiological tool you can record. So just as a preview, I'll just show you a couple of graphs and articles which we based on HIV-based studies, right? So these are some cardio-autonomic function-based studies which we used. We use time domain, sorry, frequency domain in this case.
मैं तो कॉफी दियो.
Then we have used time domain based HIV parameters as outcome variables to compare two intervention groups. So it's a wonderful tool and is easily executable with a very low barrier to entry. Anyone and anyone with a smartwatch can do HIV research today. Anywhere, anytime, any place. Right? So to quickly summarize, I'll go back to some of the resources which can be used for researchers looking to work in ECG and HIV based research. If you want to learn ECG, start with the fundamentals. The book ECG very easy. You know your basics. You want to brush up on your clinical reporting skills. The Harvard Maven database. Now you are looking for AI/ML based datasets. Get them from open source dataset repositories like PhysioConnect and Caddy. Now, if you want to use HRV, use any form of digital ECG available to you. If nothing is available, I will say a smartwatch is the easiest way you can get started, right? And the smartwatch comes with applications integrated to your phone or PC which will do your HIV analysis for you. If that applications are not doing the job, you just need to export your digital ECG either as the ECG signal itself or extracted RR signals, and these toolboxes will do the job for you. So that's my presentation. So any questions from the audience?
Thanks a lot, sir, for such an engaging and informative presentation. It's my earnest request to all the participants, please be attended because we are starting the Q&A session over here. If you have any questions to our eminent speakers, you can ask through by unmuting yourself, or you can raise your hands, and you may also write the questions in our chat box also. Joy Jayanti has raised a hand. Please unmute yourself and ask question.
Ma'am, I have a question to Dr. Arnav Banerjee. So can I ask?
Yes, please.
Sir, can you please explain what is in vivo and in vitro condition in cell culture?
Sorry, in vivo is not a cell culture. Okay, in vivo is particularly when you doing your experiment in the animal model, or in particularly any mice or rat, or in any kind of mammal cell like that, particularly the animal model. And in vitro is the when you doing your experiment outside the body. Okay, that is in vitro. In vivo is not cell culture.
Okay. Thank you, sir.
Okay.
Any
Hello?
other?
Hello?
Yes, you are audible. You can ask question.
Hello.
Yeah, you are audible. If you
Yes, I have a question to. I have a question to Dr. Vinay Chitturi.
I think there is a technical snag, so it will be better if you write the question in the chat box.
I have a question to Dr. Vinay Chitturi, sir. Sir, there is any relevance of ECG in case with pulmonary embolism? Yes. ECG can be so first of all, pulmonary thromboembolism is primarily diagnosed by from a clinical setting. There are ECG settings, which is something called RT33P3. So in the diagnostics, but nowadays modern pulmonary embolism diagnosis is based on imaging and coagulation studies of D-dimer, right? So from pure diagnostic points, ECG's role as a diagnostic tool for pulmonary embolism is not very sensitive and specific, but yes, you can always, if a screening ECG when you pick up an RT-P3 waveform, you would need to screen for pulmonary embolism. Now, secondarily, pulmonary embolism, if you are looking to study cardio-autonomic function or any altered heart dynamics. So in pulmonary embolism, in severe cases, you have to study the conditions which led to pulmonary embolism, and heart rate variability is a general purpose tool to study cardio-autonomic function and cardiac function. So both diagnostics and research. So
Any other questions from any other participants?
I have a question to Dr. Vinay Chitturi about ECG. I want to know in which conditions of our heart a single recording of ECG is useful to detect the condition, or in which conditions of our heart a single ECG is not about sure, then Holter monitoring or other tests are necessary.
So primarily when you are doing screening ECG, 12 lead ECG is bare minimum configuration, right? Because you need to do a 12 lead ECG to cover all cardiac metrics. So for diagnostics purposes, 12 lead ECG is the way to go, 5 minutes, 10 minutes. Now, arrhythmias which are episodic in nature, you would need a longer recording, right? Typical 12 lead ECG is only 10 seconds. So you need a longer recording, maybe a 24-hour recording. So in that case, can do a single channel Holter monitor recording, but modern Holter channel recordings tend to be at least 3 channels at least. Because if you are reasonably convinced that the single channel of ECG you are recording is the appropriate cardiac metric to pick up the rhythm disturbance, you can, for the purpose of diagnostic arrhythmia screening, you can use a single channel Holter, but modern day Holters typically go for 3 channels. Now, HRV wise, single channel is good enough.
Okay, thank you.
Have you any questions from any other participants? If you have question, you can raise your hand or write it in the chat box.
Hello, I have a question to Dr. Chitturi. Sir, in a clinical trial, comparing ECG screening versus no screening for atrial fibrillation in elderly patients, what the endpoints would be most relevant in these types of cases?
Okay, you are looking for the diagnostic accuracy of your clinical tool, ECG, right? So typically,
Yes, sir. Yes, sir.
So typically you would want to do what we call ROC curves and confusion matrices and sensitivity specificity tools. So typically atrial fibrillation is the gold standard to diagnose ECG. Very easy to do it. Now, typically atrial fibrillation can be also studied indirectly from imaging studies where you look for atrial chamber morphologies and changes, right? So you would need to do basically gold standard analysis things, so that things like bland Altman analysis, these kind of tools which you can follow. So if you don't mind, may I ask you what's the alternative diagnostic tool you're considering other than ECG to diagnose atrial fibrillation?
Thank you, sir. Thank you.
Any other question left to any one of our eminent speakers? Any questions from any other participants?
Leila.
We are waiting. If you have any questions, you can ask our speakers. I think no questions left from any participants. There is no questions also in the chat box. No one has raised their hands. So I think no questions left. So we are ending our Q&A session over here. Okay. Okay. So we are not wasting our time. So on behalf of the South Asian Association of Physiologists Research and Publication Subcommittee and South Asian Center of Excellence in Physiology and Research, I extend my deepest gratitude to all the participants joining us from across South Asia, India, Pakistan, Nepal, Sri Lanka, and Bangladesh. A heartfelt thanks to our eminent speakers, Dr. Arnab Banerjee, Dr. Aniruddha Roy, and Dr. Vinay Chituri for sharing their invaluable expertise. The insightful presentations have undoubtedly enriched our knowledge on clinical techniques, on fundamental research, on cell culture, animal models, and ECG applications in biomedical research, which is crucial for advancing physiology in our region. I am indebted to Dr. Kaushik Bharati for his guidance to the subcommittee. We sincerely appreciate the support and guidance provided by Professor Samina Malik, President of SAP, and Professor Naima Sultana, the Secretary General of SAP. To all the attendees, your active participation and thoughtful questions during the Q&A session was highly appreciable and made this webinar a dynamic exchange. To remember, all the participants who have registered themselves and joined today's webinar are eligible to receive a participation certificate. We hope the knowledge we gained today will empower us in our academic as well as professional pursuits. We formally conclude today's webinar on fundamental research methodologies, wishing you all the very best for your future research endeavors. I am giving the mail ID in the chat box. You can ask with any questions if you have, if any doubts if you have, to our eminent speakers. So today, good night, stay safe and stay connected. Thank you.
Thank you all.
Thank you. Have a good night.
Thank you. Good night.
Thank you. Thank you very much.
Thank you very much. It was an outstanding session. Thank you all the speakers.